Well, hello, everyone, and welcome back to our virtual event. I'm Bobby Parker from Beyond Clean. Now, before we get started with this next presentation, I want to take just a moment and call everyone's attention to the features that you'll see on your screen. If you're not familiar, all the little windows that you see around are movable. So feel free to rearrange them, shrink them, enlarge them, do whatever you need to do to make sure you get that event experience that fit your preference. On the lower left, you'll see an important window. That's the Q&A window. So throughout the presentation, we would encourage you to send in your questions for speakers to answer during our Q&A session at the end. If we do have an overabundance of questions that come through and we don't manage to get to all of them, we will make sure that those questions get to the speaker, and we'll get you some contact information as well. In the upper right, you'll see a resources window where you're going to find some additional resources to download and access that are provided to you by our event sponsor. Now, our next speaker today, Alpa Patel, is a certified microbiologist and principal scientist at Nelson Laboratories. She specializes in overseeing test method validations for reprocessing, writing standard test protocols, and standard operating procedures for reprocessing. Alpa is a member and collaborator with the American Standard Test Methods, as well as many other industry standard organizations. So as she comes to us today, we ask, why are reusable medical device cleaning validations important for hospital practices? How are IFUs and cleaning validations connected? Alpa's here to help us understand and discover why reusable medical devices cleaning validation tests are critical to a hospital's practices. She'll also identify what it takes to make a successful IFU that is sustained over time. Get ready for this cleaning validation tell-all. Let's welcome Alpa.
Thank you for the intro. I will be going over reprocessing validations, particularly in a lab setting. And like you saw the intro, I'm Alpa Patel. I am a principal scientist at Nelson Labs. I've worked in the reprocessing field for 19 years now, and I've seen a lot of things come through the lab, from little hemostat to clamps to flexible endoscopes to big machines, AERs and TEE machines and probes. And so we've seen a wide array of devices for validations, including the eye, dental, orthopedic, spine, anything you can think of, we have probably had our hands on. For the agenda, I will be going over reusable medical devices, what needs a cleaning validation, right? That is one topic that the industry still needs a little updating on, because a lot of times we do see manufacturers say, hey, do we really need this for this type of devices, or for non-critical devices that are not patient affecting? And so those are the things that I will talk about. What entails in the cleaning validation? We have manual, we have automated, IFU challenges, how are human factors considered in these validations, and trends in the medical device industry regarding cleaning, regarding IFU and life cycle. So we'll go through all this today, and hopefully after this session, you will come out with a better understanding of what it takes to perform on these validations and bring it over to the user side where they're actually performing these every single day. So when we see when I talk about reusable medical devices, I'm talking about devices that are intended for repeated use, right? It's used on a patient, then you process it, and you use it again. So it's used over and over and over and over again. What is the end point? We don't know. We're still working on that, correct? I'm sure all of you guys have seen these type of devices come through your department for cleaning, and we do validations on every single one of them. These are something that you may have not been familiar with. Do these get cleaned and validated? Sure they do. I mean, you have them in your reprocessing facilities policy to clean them, but are they validated? Do they show that they can remove blood efficiently, protein efficiently? do we really need to go to that extent? Is visual inspection okay? Where does this fall into when it comes to validations? And right now, non-critical devices, which you're seeing here, I've got dots everywhere over here. The yellow dots here, they are, believe it or not, validated for reusability, cleaning, and disinfection. Of course, they can be sterilized, so they will be disinfected, and it will probably be low-level disinfection. But cleaning is a must. It's just not wiped and you're done with that process. You actually have to assess for residuals that are on these devices. And one thing I want to emphasize is when these surgical lights, we had no idea how big they were when we got them in the lab. You see them in the picture and you're like, yeah, okay, we can do a validation on that. When it came into the lab, it was so big, and we could only fit one in our lab. So it was a challenge, but we got it done. But it gave us a perspective of what healthcare has to go through in everyday uses, how we validate things to how the healthcare is responsible for. There is a gap in that area, and to understand and bridge that together is something that the industry still needs to work on, and I think that we're getting there, though. I'm going to go over some cleaning validations that we do and why these validations are important. And all the devices that you do see in your side of the world, they are, believe it or not, are validated. And so this gives you a perspective from the lab side of what is needed for the validation and how we go through those. Now, we all know there's a point of use treatment that's performed. And then I've colored this thorough cleaning here because a lot of time this area does get looked over by manufacturers, and they're like, okay, you just need to rinse it under water, and then it goes to disinfection and sterilization. But that alone does not do the trick. As you all know, we need some sort of instructions, an enzymatic detergent or brushing or flushing. All those have to happen for the next steps to be performed adequately. But we do go through this entire process, but the point of use treatment usually is not used in the validation. And we go straight to thorough cleaning, sterilization, and disinfection. Today, my point of focus is just on the cleaning validation, but we do perform disinfection validations and sterilization validations. They are not in conjunction. They are performed separately, as their end results are different. So for cleaning, we're looking for analytical markers like protein, hemoglobin, TOC, carbohydrates, stuff that you do already when you're doing ATP tests. But ours are different. And once those are done, then a disinfection validation is a standalone validation, and sterilization is a standalone validation. So they're not done together as it would happen in a hospital. We have to evaluate them separately. Now, for cleaning validation steps, it's basic, simple. It's only 4 steps. We soil the devices with simulated use test soils. Here, we're just guessing what the test soils would be, or we work with the manufacturer to see how the device is used, the intended purpose of the product, and try to come up with a test soil that mimics the product. After that, we're cleaning it through manual or automated methods, depending on the IFU that's provided by the manufacturer. We extract those sites that we are cleaning, and then we test for the analytes. Now, before we do the cleaning validation, there are cycles called simulated use cycles that are performed. These are called repeat processing, where we are processing the device multiple times to make them into, let's say, used conditions. And what I mean by that is back in the day when I started, we didn't have this process. We would test medical devices that were straight out of the manufacturing box. It's shiny, it's pretty, looks very nice. And we did validations on those. So the possibility and the probability for it to pass is much higher than it would be if I cycle them for reuse cycles that you have to deal with every day. So FDA implemented this in 2015 in their guidance document and specified you should consider simulated use cycling before you do a cleaning validation. And when that happened, it changed the whole realm of validations. And we had to do these cyclings where the healthcare facility would see a device processed from use to cleaning to disinfection, if that's the last phase, or sterilization. And we do that 6 times to make them into used condition. And then on the 7th time is when the validation would happen. So this was implemented just a few years ago to assess cleanability. Now, you might be asking me, well, why are you all focused on cleaning validations? So why is that such a big deal? Sterilization has always taken the platform when it comes to reprocessing, but what misses in the link is cleaning, right? If you can't clean a product appropriately, you can sterilize all you want. Nothing's going to happen. You're still going to get infections. And this is an example of it. I'm sure you've seen this example in many different webinars or presentations before, and it is an image that stays with us forever. These are suction tubes that was studied at the University of Michigan Health, and this is tissue that was on the device, and yeah, it was brushed. The IFU was followed. Everything was done to the instructions for use and then sterilized. So why do we have this here, right? This question is something that came back to the laboratory, right? Hey, did we simulate the test soil correctly? If there was tissue in it, was the brushing steps performed appropriately? So we evaluated that having a corner like that, we couldn't brush the debris off, and so it had to be evaluated a different way. This picture on the right, where you see the rust happening, this is only after 6 reprocessing cycles. So thus, the FDA's concern was valid to perform reusable cycles before you do the cleaning validation. And we saw this just after 6 cycles. And this is very alarming because things were flaking off from the inside. So the outside was perfectly fine, looked polished, looked very well. But when we went with a boroscope inside, because things were failing, so we're like, why is this failing if it looks really good? We put a boroscope in the channel, in the lumen, and this is what we found. So we took pictures and sent it to the manufacturer and said, hey, we have a problem here. If this is happening after six cycles, your device needs to be redone. It needs to go back to the drawing board, and new materials have to be used for manufacturing your product. So besides all of that, you might say, what are the other things that we do for cleaning validations? We have strict controls that we have to have, and even replicates that we have to perform. So this whole screen here you're seeing right now are all the controls that we have to perform to validate the validation. I'm not going to go in detail on all these controls, but I just wanted to give everyone an overview of it is a validation where we have a positive control, negative controls, test article controls, and cytotoxicity control. This is where we're evaluating patient safety regarding detergent residuals. Are there any residuals left after cleaning the device that may affect patient safety or subsequent steps afterwards? Because there may be some detergents that are not compatible with the disinfections or sterilization. So those are the things that we evaluate during our validation. We also, these are some of the outlines of the controls we have. If you think about it, the main control we have is a negative device control. Now that control is a control that we clean the device, right, without any test soil. So it tells us if there was any residuals left on the device that would change the result. Positive is where we put the test soil on it and then we extract it. So we know what the device had in the beginning to see how much we were able to remove in this in the cleaning validation. And the other controls, the sample controls, are for the assay that we use to evaluate the protein and the hemoglobin to make sure that there's no interferences that is affecting the test result. So all of these controls put together helps the validation solidify and gives the results confidence that these results are very accurate for the validation. Now you might be like, okay, what kind of soils are you seeing? Because it might not be the same soils that we're seeing, right? So we do use simulated use soils. We're not using native soils or human blood. We do use blood soils that are derived from animals. And there are many, many different soils outlined in the ISO documents and in ASTM documents that we do use. We do have coagulated blood soils. We do have blood soils that are not coagulated. We do have mucus-based blood soils. If you see up here, this is called a British test soil. That's mucus-based, right? And then these blood soils, and you can see that this is a coagulated soil where it just clumps up like that. Now, I have a question mark here, and that is because a lot of orthopedic devices, like we saw a picture of where it retained the soil, they might have tissue, or they might have bone meal stuck in there, right? How do I simulate that? Because this doesn't have it. We add things to the test soil to ensure that we are closely mimicking. It's not going to be exact to what the procedure will see, but it's close enough where we feel confident that we are challenging the device enough. So these are some of the simulated test soils that we use for the validation. Now, can we use any test soil? Probably not. So FDA does have an outline that any test soil you use to scientifically justify the test soil and show how it's clinically relevant. And what I mean by clinically relevant is we have to show that the test soils that we're using is what the device is seeing at the end, after the procedure has been performed, and what the users are seeing to clean. So the regulatory does make sure that the testing labs, when they're doing these validations, are working with the manufacturers closely to determine what kind of test soil we should be using for the validation and mimics the actual practices that are happening in the healthcare facility. Now, I'm not sure if everybody's aware, but when we see a tray like this coming for a validation, we're not testing the entire tray. We do select a worst case device. And how do we select a worst case device out of this tray? There is probably so many instruments in there, and we would have to pick what we with the manufacturer think it's worst case. And the idea here is that if you choose one or few instruments out of this tray that have the worst case features, designs, lumen size, and if I can clean that with the instructions for use provided by the manufacturer, then I can clean the rest of them because they fall in that umbrella for testing. So in this case, I'm just giving an example. In this case, we would pick this because it's got the knurls on here. I can open it up and close it up. So all this stuff is inside, is contaminated. It's got all the design features that are very difficult to reprocess. So if I clean these, I should be able to clean the rest with the same instructions for use. Same thing here. This device looks very complicated. It's got all the design features that will be very, very difficult to reprocess. So we would pick this type of device and try to clean what we can and challenge this device with the test soil and procedures. And then this would be used as a worst case device for the rest of the product. Family grouping is another common factor used for cleaning validations, where if you have a whole area of instruments like that, these are drill bits that look exactly the same, but the ends are different, the lumen sizes are different. We would choose just a few from the family and say that if you can clean that, then you can clean the rest of the devices. Of course, you have to choose what is more challenging to clean, where you're looking for the smallest lumen size. You're looking at the end bits to see what will retain the soil the most. Those are different. And then when we get non-critical devices or even semi-critical devices like a flexible endoscope, we're not testing the entire device, but we're selecting test sites for it. Of course, with the flexible endoscopes, you guys have heard there's a lot to worry about. And so we do challenge the endoscopes quite heavily. And they are good size channels that we do test. I mean, we have the instrument suction channel, air water channel, control handle, distal tip. If it's got an auxiliary water channel, we would do that. So there is, all the channels would be challenged, including the exterior sites that are mostly handled. When we select sites, we're looking at what does the surgeon or what does the clinic physician contaminate during surgery? What is most contaminated? And those are the areas that we would challenge the most. The distal tip would probably, I mean, it might not go all the way, but we'd probably test it all the way up to here. So it goes all the way. And so that is how we would pick sites. And same thing with the picture on the left, where it's monitoring the heart rate. We would have the on and off switch, the top of the device where it's handled when you pick that up, the sides. So all of those test sites would be tested individually and evaluated for cleanability. Now, when we have selected the sites or the worst case devices for testing, now we're working with the manufacturer to figure out how are we contaminating these? What are we flushing them? Are we soiling them with contaminated gloves? What are we doing? So all of these things that I've outlined here is what we are discussing with the manufacturer to figure out how this device is used and how can we actually mimic the process that's happening in the healthcare facility to actually make sure that the cleaning validation is done accurately to what is happening at the facility. And simulated use, where we're figuring out how long is this device used? Is it 15 minutes, 20 minutes? So we know what the time is for soiling. What is the wet contact time? How long is this device used in the surgery? And then the dwell time. We're always looking at the dwell time, right? Once the device is done, it's put on the side. How long is that device sitting before it goes to the central processing for cleaning? That is also considered in the validation. This gives you an example of what I'm talking about with simulated use soiling. This is how it is actually being used, and this is the amount of test soil we're using, and we're simulating, right? This is my hand with gloves that are contaminated with test soil, and I'm holding the scissors, and I've actually soiled it more than what you see here is because to challenge the processing, just in case. If the scissors get very soiled, then how can we clean it? So we have to think about worst case scenarios when we're doing these methods as well. We will soil them to way worse case than what the procedure would be contaminated with, as you can see here. There are several different ways or methods of soiling. We have immersion. So if it's going directly into the body cavity, we would immerse the device directly into the blood soil, just like we have the hemostats here. If it's a spray, we would take a spray bottle and spray the devices. And the spray would simulate if you're cutting something in the body and it just sprays on out, then that is what we're simulating. The handling with soiled gloves, which is a common practice that we do all the time. And then in the hidden background, you see here, we have a flushing on the channels in the endoscope. And so we also do flushing as well. Any lumens that are on the device will be flushed with the test soil to challenge the lumen cleanability. Now, dry time validations. Back in the day, when we did validations, when I first started my job at Nelson Labs, we would have a wet towel with water, and we would just place it on the devices for 30 minutes so it would keep moist, because those were the actual procedures that were happening in the hospital. So to mimic that, we did that. Over time, things have evolved and changed when it comes to these validations. And we are trying to mimic what is happening in the actual facility now, right? You want to make sure that you are challenging the cleaning as much as you can. So it has come to the point where all the validations now are performed where the test soil is left to dry at room temperature. And once it's bone dry, that's when we will start the cleaning validation. So we're challenging the processing. And the reasoning for that is, if you see this picture here, we got this from one of our customers that was working with us. And they told us that when we're doing these validations, devices sit in these trays, right? And soil can seep down, or it's sitting there but you have to get these cleaned in an 8-hour period, but they can be dried before they get to them. So that's why you want to challenge the process, where if the device is dried, the cleaning instructions outlined in the IFU can still be feasible and will get the device cleaned. So we do challenge it till it's that the test soil is dry. Now, for the validation parameters, this is your manufacturer's IFU that you would see at your end. The validation parameters is what we would do on our end, where we would challenge it to the lowest dosage. We would soak it to the lowest time. We would flush with less detergent, and we would use the temperature that is specified to the lowest. And this is what we would challenge for the processing, just to make sure that if time is not permitted and you have to do it at 2 minutes instead of 5 minutes, the 2 minutes is validated. Because 5 minutes, if less is validated, then more is fine. So our validation procedures always go to the worst case process, where we would go on the lower end of everything. Some of the manual cleaning methods, we have manual cleaning through brushing, which you guys see. And there we also have faucets that healthcare facilities have. So we have designed our labs to mimic what healthcare has, just to make sure that we are doing what we can to make sure that we are closely representing what healthcare facilities have in terms of cleaning. Sonications is smaller. I mean, we don't do it in large amounts, but we do have sonication bats to simulate any ultrasonic cleaning that's required. And those are usually used for orthopedic devices. Not every device goes through ultrasonic cleaning, but if your IFU does specify it, then we would do it. Just an example of our test lab setup. This is our 3 sinks, and this is where the instructions are posted when we're doing our validations. You have your instructions when you're doing your cleaning, and we have our protocols that are specified for cleaning, and they're outlined all the way here. So an analyst is standing and reading each process and doing cleaning here, rinsing here, drying here. So our labs are also set up in a way where it mimics closely to what the healthcare facilities are seeing and are using as well. Accessories used for cleaning. Same accessories. We also try to keep the accessories similar as much as we can. Lint-free cloths for everything that dry. We don't want any particulates. Syringes, brushes, scoring pads. This is only used if there's electrosurgical procedures that have happened where the test soil has baked on the ends, where you can't get it off with a brush no matter how many times you're going to soak that. So you need to have a help of a scoring brush to make sure, but These are also not an all the time thing we do. This is only specified if the manufacturers specify in their IFU. So a lot of the supplies driven is through the IFU. Now, if the IFU does not have this supply specified and we think that we need it for the validation, like ultrasonic cleaning is another one that a lot of manufacturers do not specify, but if it can be cleaned manually without the ultrasonic part of it, then we would add that to it. Some of the brushes for the lumens. And then methods of automated cleaning. As you can see, these are automated washer disinfectors that we also have that we use. We have Steris, we have Belameds that we use and process them in the trays, just how you have them. For the validation, we do not perform thermal disinfection. That step is missed and not used as it does affect protein. And so we only go through this process and then do the validation. Detergents, we have to ensure that the detergents are compatible to the end user. Not always do we validate. That's one thing that there's a huge gap. And what we validate is not something that the healthcare facility may use. So we always want to use something that is available to the end user. And sometimes it is available, sometimes it's not. So we work very closely with manufacturers to ensure that where they're selling the product and who they're selling it to, we closely mimic what detergents are used over there, including Europe. Visual inspection, as it's done in the healthcare facility, we also do it through naked eye. Just looking at it, that's our first. Second is under magnification. These are for complicated devices or duodenoscopes that we're validating to see the distal end portion of it. Borescope upon request only. We don't do it for everything because it's not required. So if the manufacturers do specify that we want you to go in and look for stuff, then we would do. But otherwise, it's 1 and 2 are what we would use. Once all that is done, we've cleaned the devices, now we're extracting them. And there are different ways of extracting devices. Like you can see, we flush if there's lumens, there are manual shaking, if there's computer screens or non-critical devices like we saw in the beginning of the presentation, we would use swabbing. So there are methods of extraction that we would do. And once they're extracted, we do test for analytical markers, protein, hemoglobin, total organic carbon, all of this stuff is what we're looking at when we're doing these validations. We do have acceptance criteria that we have to meet per standards. If they don't meet these acceptance criteria, you fail the device and you have to increase your cleaning procedure to meet the acceptance criteria. Nobody's going to go redesign because you're at the end of your phase when they're doing these validations. Often we see people just increase the procedure. So when you see long procedures for cleaning validation, it's because we didn't meet these acceptance criteria for ISO and AMI. And that's what makes them longer. Is we have to add now more flushing or more rinsing or ultrasonic cleaning step to meet these. So there is a criteria for them and we have to meet them. Our instructions for use. Human manufacturers play a huge role in instructions for use. They are the first people that we have to worry about. Users are most important in this. We have to make sure that they understand how the instructions are written. And these tests are performed as well with the users. It is almost not a requirement, but highly recommended that you do perform human factors testing during your design file or your design phases of your devices to get feedback from the users. One of the things that when we did some investigation, some of the things that we found is that there is a gap here, manufacturer to sales team to healthcare facility, but the SPD personnel doesn't get the full information, right? It's like a telephone. You go from one person to one person, and by the time you get to you've missed half of the stuff that you should have known. So it's very important that training and visual demonstration of how to use should be done with the SPD personnel. And they are the first person in charge of all of this. So why are we going through this chain when we should start right here? Believe it or not, AMI-TR 55 is a standard that goes over instructions for use and how they have to be laid out for the users. And they have a font size. They even have what you have to use and how you should write. And you should be using 1, 2, 3 and short, no paragraphs. So it goes into quite detail on how you should be performing or actually writing these instructions for use. And often validation parameters is what we validate, but this is what is usually written in your instructions for use. So I just wanted to emphasize that what we validate does not go in the instructions for use. It's what the manufacturers already have. We're just validating to the instructions, but it is to the less, we're using less dosage time and stuff like that for it. Simplify information is one thing that the regulatory has emphasized, where you see these paragraphs, your eyes will walk away from that. So you want to put them in a chart form, bullet point, simple, one-step instructions. So very important to have it in this form. So you will probably see more and more IFUs come out in this way instead of that. And you'll also see for automated cleaning, instead of verbiage, they are set out to do tables like I showed you the table earlier. You just put that table in there, so it's easy. Your eyes are drawn to the table and you know exactly what parameters to use instead of written in verbal. Also disassembly, reassembly, very, very important in IFU. Regulatory agencies have asked to put pictures, some sort of illustration, so it's easy to see, just like this that I have here, right? It shows you simple how to do it, and so there is no misunderstanding. The user knows exactly what to do, how to disassemble and put it together, so we're not destroying the product or forcing something in that doesn't need to be forced. So those are the things that IFUs are making sure that they have these instructions in there and it's clear enough for the user side of things. Now, trends in the industry. What we have here is the manufacturer, we have healthcare facility, the users, other organizations, these are standards, and third-party testing laboratories. We're trying to bridge the gap between all of us together. It's going to be a while. The focus is on harmonization practices between all three parties to make sure that we understand each other's work. And when we're setting these validations, we are setting it as accurately as we can to mimic exactly what's happening in the healthcare facility. So there is a lot of emphasis on harmonizing practices. With that, there are many, many standards in the last 5 years that have started to pop up regarding cleaning validations. Just cleaning validations, not even disinfection or sterilization, just cleaning validations. And things have updated, like you can see, all 2018, 21, 2020. SD98 will be 2022, right? This is a big one for cleaning validations that's coming out. This is being it's already updated, but it will be published next year. ISO 2021, another 2021. So you can see all of these have been, the last few years have been updated heavily to influence the cleaning validation and make sure that we are doing exactly what we should be doing for these validations. One thing that is also coming up is for non-critical devices, visual inspection is something that is going to be accepted. So we don't have to test for any other analytical markers. Protein, hemoglobin, you just have to wipe and say it was visually clean and we're good to go, which helps manufacturers and also healthcare facilities where we are not adding procedures. When we do non-critical devices, we're adding so much wiping that we destroy the product at the end because we're trying to get to the acceptance criteria. And you saw the list that I showed earlier. if you don't meet those acceptance criteria, what happens? You beef up your IFU. And for visual inspection, for non-critical devices, you usually just wiping and you're done. But that alone will not take care of the analytical markers that we're testing. So you would have to wipe basically continuous for 5 minutes to get that. Who's doing that? It's not practical. It doesn't happen. And do we need it to that specificity? Probably not. So visual inspection has come out in all these documents, and it's something that will be implemented. There are more cleaning validations where more requirements where human factors are going to will be implemented for testing, patient safety requirements, more cytotoxicity testing, questions surrounding life cycle testing, right? What is the end process? When can I throw this device away? And heating, cooling systems, dialysis machines. We're seeing things that we never saw for cleaning validations. Systems that disinfect the area. How do you know those systems are disinfecting the area or the air? Those are the things that we're starting to see now that are just not medical devices, but outside of those realm. And more stringent increases for the validation itself. We have more controls. We have those simulated cycles that I talked about, and changing on how to report results. So there is a huge impact that's happening for the validation realm, and which is good for the patient side of the world and users as well. With the life cycle, this is my last slide, but for the life cycle, I just want to bring up that there's a lot of discussion happening because there's not much information out there. At what point do you stop? IFUs usually say if you start to see corrosion, pivoting, rusting, or if there's any problems in the device over here with discoloration, if this broke off, contact the OEM, right? And they will take care of it, or you go send it out for repair, send it out for refurbish. But how does the manufacturer know that? They usually have to test something about it to give you those suggestions. So it seems like we're seeing more and more testing on this because the manufacturers want to know at what point does this device start to break off, and at what point is it you can't use it anymore because patient safety is into effect. It's just not the device itself anymore. So those are the things that are coming up, and we are seeing more and more testing on it, and cleaning validations are getting very stringent and very tight when it comes to validations, which is great. And that brings me to some references I have here. These are all the standards that are used for validations. I'm sure you guys have seen some of them in your facility that you have to follow. But that takes me to my end of my presentation. If there's any questions, I would be happy to take any.
Yeah, thanks for that awesome presentation, Alpa. We did have quite a few questions pop in through the course of your presentation. So to get things started here, one question that came through is, and this is, I'm sure, one that you're going to resonate with, why can't we standardize the IFUs for all devices and or instruments no matter who the product belong to? You have some that are very generic and others that are very complicated. Now, I'm sure in your experience, you've seen a pretty wide array of reprocessing instructions. So what are your thoughts on that, and where's this industry moving in that regard?
The industry is moving towards that. We have TIR 12/2020 that did get published last year in December that do have instructions for manufacturers. We have 3 annexes that go over that if you have a scalpel, why do we have 18 different instructions to clean them? Why can't we just bring them into their buckets that we put these medical devices in, and we just follow these instructions? So we have started that. It was published last year. Manufacturers are considering that. We won't see where everything will be harmonized for a while, but we are going in that trend, and we are seeing that it is being in the standards. So it is important that we are following these standards as well.
Great. Well, I have a question.
Sure.
So you touched on the harmonization of, between the manufacturer, laboratory, and users. And I think that that's a really good topic because it's new in the sense that it really hasn't been rolled out yet, but I'm sure that it's been in process for a long time. As far, could you provide some insight on your end on the steps that are being taken and processes that are helping to really get that off the ground for the industry?
Absolutely. This is where the standard committee platform takes a role. We are heavily involved with AMI, ISO, ASTM, and in those standard committee meetings, we have user representation, we have testing lab and manufacturers all sitting at the table talking about how these standards should be outlined for validation. And many are for users, and users are there represented to say, hey, you guys are talking about this, but we can't do this. For example, AMI ST79, when we had that sonication bats should be drained every single time, every device that's tested. You drain, you fill it back up. And the users were like, that is impossible. That is never going to happen. We can't do that. Are you asking us every device we put in, we have to drain and refill? There's no way. And the manufacturer was very keen, like, no, that has to happen. That's how we validated it. So there is that. So they are quite heavy debates that we do have, and users did say that, no, it's not going to happen, and we didn't add it in the ST79. It was an 8-hour shift, right? And that was what was placed in the standard. So we are harmonizing the standard platform, at least, where we can talk to each other and figure out platforms like ISHM, where it's for user-based. We have manufacturers, we have testing labs going in there talking about studies they perform and standards that are coming. So we have different platforms that allow us to talk and interact and see how we can collaborate more. And I think Beyond Clean, this is a great platform for users and people like us, testing labs and manufacturers, to come together and understand each other's work and roles that we play for patient safety.
Yeah, great answer. I think, your point about the ultrasonics, and I mean, I ran into that, on the vendor side as well, because that was a common question. How often should we be changing out the chemical? And, my answer based on the standards was once per shift, because they were getting some guidance that were, just as you said, after every instrument tray. And Bobby, this, it's literally impossible to do that. Yeah. I mean, that's the time factor that goes into that is really, just not realistic from a clinical standpoint.
Yeah, no doubt. Alpa, how do you think that harmonization of manufacturers and users in relation to IFU is going to impact the dental world?
We do see a lot of dental instruments come through the lab, and I know that for cleaning, a lot of the instruments does go through sonication. So when it comes, when we see it on our end, we know that the dental instruments are usually cleaned through sonication methods, ultrasonic cleaning, and so we do subject them to that. It would go through a rinse and then go through that. But we're working with just the manufacturer to understand those processes. I know that because when I go to my dentist, I'm watching what's happening around and making sure that, this is what they're using. I see. And so I'm learning through that as well. But our interface is with the manufacturer, right? 90% of the time. We're not going to hospitals or clinics to see how they're doing it. We get that experience through Beyond Clean, from ISHM, from AORN, from all of these different platforms that we get involved in to learn. But for the IFU, it is difficult to make it so it's the same throughout. But I think for simple devices, it should be harmonized very easily.
Yeah, thanks for that. One follow-up question I have on the testing procedures. You'll sometimes see IFUs that have very specific products in them. Maybe they'll call out their enzymatic detergent by brand name and product name. In those instances, do you have to use those exact products or one that's just a comparable makeup? How do you handle that on the validation side?
On the validation side, when you see the name written on it, we are usually using that name for the validation. Now, for the user end side, they usually, when we use a brand, I use Enzol detergent, right, for the cleaning validation. It's an enzymatic detergent. The manufacturer should be writing a neutral pH enzymatic detergent. No brand names, just that. Yep. And usually they do work efficiently if they are in the same category. So if it's an enzymatic detergent, they should be working efficiently. They are different enzymes in different detergents, so that has to be thought out as well. But that is where the gap is. Like I said, what we use for validations might not be what healthcare is using for validations. So are these validations even relevant at that point? Because we're using two different things, but we're using the category enzymatic detergent.
Thanks.
Yeah, that's a great point, because I've had interaction with facilities where in the IFUs it states a specific manufacturer of detergent or a specific washer, which is even more unrealistic because given the expense of of washers and all that capital equipment. And I think, I've had the phone calls where they're like, I don't know what to do because it's telling me I have to use this specific brand. And so I've taken the step forward and guided them, saying, okay, that is an enzymatic, or that's a non-enzymatic. If it's a neutral pH, you should be fine, because that causes a lot of confusion. It does. Makes them think that they need to have that specific brand, and then all of a sudden everything stops. So yeah, very good point. I think that just mentioning not the brand, but the type of detergent or type of sterilant, however. So question, when it comes to validation, are you doing ATP testing only, or a controlled bacterial culture growth test as well?
So for cleaning validations, we don't do any microbial testing. That is not required at all for cleaning validations. Okay. But we don't do ATP testing because we have to do quantitative assays, fully quantitative assays. So we have to have a limit of detection, limit of quantification for the assays that we perform for hemoglobin. ATP is not that precise. That is for the end user to use, just a quick test to see if it's there. If there's blood or if there's protein, then I need to put it back into processing. For us, we have to have all our methods that we're using validated and have strict guidelines for ICH documents. So those are very different from what the user is using.
Great answer. Thank you. Yep.
This kind of branches off from our topic a little bit, but you brought to mind mentioning end user testing. You talked a little bit about visual inspection, but do you have any recommendations for end users out there of the science of cleaning verification at the user end? You mentioned ATP testing as one, but there's a host of options out there.
There are a host of options out there. There's many that I know HealthMark has many that you can use, where it's a hemo check, or you can use a swab and it will show you a color change. There are many different avenues that you can go to find these tests. But with chemical assays come with interferences. That is one thing that we as an industry haven't touched, is if you have any residuals of detergent left, it can change you think you've got blood, but it's just detergent. It's a colorimetric assay, so it's going to color it if there's any pH change. So those are some of the factors that we as testing labs know, and but that have to be evaluated, but we haven't gone there yet. And that's why we have to use analytical methods to make sure that what we are testing is accurate. And the test kits that are available for end users to use to make sure that there is just a quick check for them to see that, hey, it's clean, there was no color change, we're good to go, and move process. So there are different ways of doing these validations, but there are many out there that you can definitely use.
Sure.
Here's a good question that came through, and this, I think it merges a little bit with the harmonization aspect, but when you can't branch out and you can't get in clinic and use an end user or have focus groups, that type of stuff, do you incorporate human factors when reprocessing devices so that your lab processes more closely mimic what really happens in sterile processing?
So we try to do it that way. Like I showed you our lab setup with the 3 sinks, with the faucets. Now, if you're asking us if we're wearing the gear, we're probably not wearing the gear. We're not wearing the rubber gloves that are all the way up to here. We're not wearing the face shields. We do have masks. We do wear masks, but we don't have hats. We do not, our laboratory is controlled environment. So many times we hear that it's too hot in the SPD departments. We usually don't have that issue. So there is that gap. I mean, we're not wearing the gear that they're using to reprocess. We have our lab coats on, we have our face mask on, we have our gloves on, but they're not the heavy duty gloves that they're using.
You mean you don't have stacks of case carts coming out of an elevator that are...
No, we just have one device type that we're validating for that day. So it is different, right? Yeah.
Oh, for sure. Yeah, it's really hard to mimic exactly what goes on because, as Bobby knows, when you're in a department and going throughout each day is different. The surgical cases are different. It's literal chaos sometimes, and it can be very stressful. You're on a time limit to get to the next to deliver the tray for the next case. And you're also really wanting to try and focus on quality, but speed is always an issue. And I know that's those two have been discussed in many instances. So, Bobby, you have anything else?
Yeah, Alpa, in a previous conversation, you mentioned some of the contrasts between validation that takes place in a laboratory setting and then those facilities that are conducting device validation or process validations kind of at the hospital. Do you want to give us, maybe some quick distinctives of what separates those two things and how we're kind of slowly trying to move them closer together?
So some of the things that separate them is the instructions for use, right? That's a big one. What the users are using for instructions for use is specified in the IFU. This follows step by step. We're following step by step. However, we're taking some steps out. We're flushing with 40 mils instead of 60 mils. We're making detergent baths at a quarter ounce instead of 1 ounce. So that's a big change in there. Another thing is like. You just mentioned we don't have the stresses that the SPD departments have. We are working at our pace. We have one product that we're focusing on for cleaning, so it is not stressful as it would be in healthcare. We're not timed, right? If you would come see how we do our validations to what it is different. It is not the same thing. So those are some of the differences. The gear we wear is not the same as well, but it is some, and I can't say it is the same thing because what the users go through is probably 100 times more stress and focus. And they're like you said, there's carts and carts coming. We're not worried about the carts. We're only worried about that product that I'm validating for that day.
Yeah, well, thank you for sharing that. We are coming in at our 1 hour mark, so I think at that point we're going to go ahead and close up the presentation. I do want to thank you, Alpa, for sharing your professional knowledge with us on this really important reprocessing practice. If any of you have any questions that we weren't able to get to or you didn't get the opportunity to ask, you can connect with Alpa via email and she'll gladly answer them. Do you mind to put your contact information there on the last slide back up for our participants?
Oh, yes. I don't know how this went over. Sorry.
No, it's okay.
Yes.
Now, we're going to go ahead and take a short 15-minute break before our next session. So thank you all so much for joining us. We will see you for a discussion dedicated to navigating change in sterile processing very soon. Thanks, everyone. We'll see you here in about 15 minutes.
Thank you, everyone.
Thanks, everyone.